and also really thinking about good clinical endpoints, because as we'll talk about, DUP15Q is a condition that we've been studying and treating for a long time, but it frankly has a lot of features that are consistent with many other neurodevelopmental rare disorders. So epilepsy, motor impairment, global developmental delay, things that we need to think about in other conditions too. I'll talk to you quickly about what we're doing in TSC as well. So these are the three kind of, again, initiatives. I love acronyms for studies and cute logos that my team makes. So the DUP15Q stuff is under the umbrella of a study called BBIGS, Brain and Behavior and Genetic Syndromes. Okay, so we all know about DUP15Q. I don't have to go into the detail here, but as you know, these children often have not simply autism, which is highly penetrant in this condition, but also global delay, intellectual disability, and hypotonia. You've heard a lot about the genetics, we won't get into it, but just that in addition to UB3A, which has been the focus of much of the research in this condition, that there's many other genes in the region that are variably overexpressed, and we've been interested particularly in the 3 GABA receptor genes. And why is that? Well, because Larry Reiter and his group wrote a paper many years ago where they were characterizing the interstitial, children with interstitial duplications, and they were describing the phenotype and other things, and they commented on the fact that many of these kids on their clinical EEG had this signature. There's these fast oscillations that seem to fall. They didn't say fell in the beta band, but they were these fast oscillations that do fall in the beta band. But they look like, it looks like the EEG of a child or an adult who's taking a positive allosteric modulator of GABA, so a benzodiazepine. So when I see an EEG like that as a neurologist, I think this person's taking medications. They're taking Ativan or Valium. Very interesting, given that GABA, we know that GABA neurotransmission is altered in this condition. We know UB3A modulates GABA co-release, as well as these GABA receptor genes that are overexpressed, again, in variable amounts, probably affect the way GABA signaling is actually happening. And I say that somewhat vaguely because we don't know actually that much more about what exactly is altered in GABA neurotransmission. But again, this EEG signature suggests that something is going on. And so that became very interesting to us in my lab because we do a lot of work with electrophysiology, studying biomarkers across different neurodevelopmental conditions. And this was such a robust signature. We're searching for EEG biomarkers in infants at high risk for autism where the signal, the variability is so subtle. And here we have this signature that is it's so extreme that you can see it across a room, literally. So we asked first, can we actually quantify this? And again, you've heard this for any of you who've been in these meetings before, so I won't get into it again. But we asked, can we quantify it? And if we compare these kids with DUP15Q to non-DUP15Q, is it really higher? What part of the, where on the scalp are we seeing the signature? That sort of thing. So we collected a ton of data at the conferences as well as at UCLA. And what we found, and this is published, is that in fact, there must be a pointer on this somewhere, one of these, right? Yeah. Is it the red thing? Wait, which one is it? Oh, this one. Yeah, okay, good. So this is just a heat map. You can see DUP15Q. This is just in the beta band. Beta is much higher in DUP15Q compared to IQ-matched kids with autism, as well as typically developing kids. Power spectral density plot, you see this big beta peak. And the effect size here was almost 2. I mean, really high effect size for an EEG finding. So since then, what we've done, and again, I'm doing this quickly because I gave a full talk on this last year, and this paper about the properties will be out soon. We've been since then asking about the properties. Is it stable over time? The answer is yes. Is it scalable, meaning can we quantify this in clinical EEG the way we can on our research EEG? The answer is yes. Does it correlate with phenotype, which is the question I always get asked, and it drives me crazy because I never thought it really should. I think this is really a biomarker of the underlying, sort of genetic aberration, if you will. And so to think that it's going to relate to something as, I think, downstream and crude as IQ, to me was not that compelling. But everyone was asking, so we looked, and it doesn't. The peak at which the beta occurs is actually very different in kids with and without seizures, and that's something we're further exploring. So it does seem to be a biomarker that relates more to whether or not a child has epilepsy. But really in terms of phenotype, no. And then we asked, as I mentioned in the other room, if you were there, does it change with state? So I was just curious if beta persisted in sleep, really because I thought if beta persisted in sleep and it altered sleep physiology, or kids didn't have sort of typical sleep physiology, that might relate to cognition and other neurodevelopmental sequelae. And I'm not going to show the data today because it's really early and we want to publish it first, but the reality is we do see beta in sleep to varying degree, but as we started looking, what we found was that sleep physiology is in a way that I've never seen actually in a condition. It is incredibly altered in DUP15Q. We are seeing incredibly, and I'll use the word abnormal, just abnormal sleep physiology in these kids. So I just got an R21 to quantify sleep physiology in DUP15Q, and that's why we're asking the parents to give us all their overnight EEGs so we can quantify this better. But another question that we wanted to ask was, what can we infer the underlying genetic mechanism of these beta oscillations based on clinical data? So really what we want is to look at this in the preclinical models, and there's folks doing that, and we haven't, that hasn't been published yet, but there are folks, I know that Toru is working on it, as well as Payman Golshani in our lab and many others, to ask whether, does the UB3A overexpression mouse alone show this signature, or do you need the other genes, right? So we thought, well, how can we, with clinical EEG, at least ask the question about whether UB3A is necessary, overexpression is necessary for this, this EEG phenotype. So this is a paper that just came out, and this is a graduate student of mine, Joel Froelich, who worked with our team as well as some folks at Roche. Jörg Hipp was the physiologist there. And we did a couple of things. So the first thing was, which I thought was really creative, was that they asked, well, can we compare, using our same signal processing techniques, can we compare the EEG of individuals who've received a positive allosteric modulator, midazolam, which is a benzodiazepine, can we compare that signature to DUP15Q, and do they look really as similar as we think they might? And that, again, just starts sort of suggesting that the GABA signaling is important, at least. And so that's exactly what they tested, and what they found was that, so this is a figure of, it's healthy adult controls, actually, who received midazolam, and this is right after administration, and what you see is this big peak, this is the power changed from baseline to receiving the med. You see this big change in the beta band, and particularly in these sort of frontocentral, really central electrodes, what's interesting is the peak frequency at which this beta occurs is at 23.1 hertz, which is the exact same peak frequency, which is where we see the beta oscillations in our DUP15Q cohort, which was pretty striking. This is just the comparison of our DUP15Q cohort, right? So here's the 23.1 hertz, and this is the GABA administered adults. We also, we sort of thought, well, how else could we examine this? Well, we thought, well, let's look at EEGs in kids who have paternal duplications, right? So these are paternal interstitial duplications. They don't presumably have the maternally imprinted UB3A overexpressed. And we thought if we see high beta in these kids, it suggests that UB3 alone is not sufficient. Like the other genes actually are contributing to this phenotype. And what we found was that, in fact, the kids with the two kids with the paternal interstitial duplications do have high beta. And you can see here, this is like these are just the channel level data. This is the beta peak. This kiddo still did have a beta peak these are all the DUP15Q kids. This little yellow dot is where this interstitial sits, and this one sits way up here. So they're still in the mean of the DUP15Q group. So again, suggesting UB3 alone is not enough to really explain this phenotype. Okay, so that was the EEG, and again, that paper is published, and then the properties paper will be out soon too. We've also looked at outcome measures, and again, thinking about what outcome measures would we want to use in a clinical trial. Really the question is, how do our standard measures behave in this group where we know there's a range of cognitive function but pretty severe intellectual disability in at least a subset of our kids, right? And so the team here of this, we did testing actually at last time's family conference as well as again at UCLA. And when we did cognitive testing, we used a bunch of different measures based on the ability of the child. What we were finding was, when we looked at standard scores, right, the normed rate scores, these kids all fell below a large subset fell below what we would consider the floor. And so the problem with that is that it doesn't mean they don't have wonderful abilities, but our tests are not able to pick up enough individual differences. So imagine, how are we going to use those scores then to measure change over 2 months of a clinical trial? So one thing that Charlotte and her team did, and Charlotte's next door giving a talk later, is they were creative with how they looked at the scores, and they created ratio IQ scores, which is is taking the age equivalence of the child over their chronological age. And that gives you a little bit more granularity in the actual scores. And when you do that, you can see, so this is the ratio score of cognitive ability, and these are kids with IDIC, with and without seizures. You'll see that if we had stuck to the 40, we would have only had data on these kids up here. Everyone else would have looked the same. If you use ratio scores, you actually can see some granularity here that is helpful because it might be a way to get around some of the issues in the standard scoring and allow us to maybe get some, again, sensitivity to individual differences and maybe some subtle changes that we can detect with intervention. We also looked at the Vineland, which I really like. It's a great measure of adaptive skills, and it's a measure that pharma companies also really like a lot as clinical endpoints. And the good news is the Vineland behaves really nicely in this condition without having to make any changes to scoring. We see pretty nice variability. This is just within our kiddos with isodicentric duplications, but with and without epilepsy, that detail doesn't matter. But we do see a nice range, and you can see this shown here a little bit more clearly, where frankly, even with the ratio IQ, we do see kind of a leftward distribution, if you will, of the scores. But again, remember, if we cut off at 40, we would have all these kids would be in one cluster. So the ratio IQ helps us. But with the Vineland, which looks at adaptive skills, we do see a pretty nice distribution. So it's a measure that we're going to keep using. We like it in this condition. We've also looked at motor skills, and again, in the interest of time, I'm not going to show you these videos, but I was going to show you 2 videos of kids who have very different levels of functioning motorically, but they score exactly the same on some of our standard measures of motor function. So I have a junior faculty in my group who's really interested in looking at quantitative motor skills and has been looking at things like the gait mat, which we actually brought to the meeting a couple of years ago, where kids just walk on the mat and they can quantify various features of gait. And you can see, like, this is what a kiddo with non-syndromic ASD looks like, a bit narrow-based, and with DUE15Q, this kiddo has a much wider base gait. And what they're finding on quantitative gait analysis, she has not submitted this yet, but is going to soon, is that looking at a couple of variables, this is velocity of movement, and this is the step length, that kids with DUE15Q do have, or look different than kids with non-syndromic autism. But the key here is that we can quantify this feature in a way that gives us, again, a little bit more stratification and a bit more of an objective way to measure motor skills. So this is another tool that we're using, again, when we think about outcome measures for upcoming trials. We're also looking at sleep, and I'm going to talk more about sleep tomorrow, but I'll just say we just finished a study where we were asking whether our traditional measures of sleep behavior, and we're looking at the CSHQ here, how does that measure also, again, behave with this group? Meaning, are they skipping a lot of answers? Are we getting any signal with this? And it turns out, and actually Jamie Barstein is here in the group, she's been working on this, on these data. Turns out it actually is pretty good, and parents are able to fill out most of this and give us good data. And we do find, again, the actual scores I won't worry about, but just looking at the different subsets, we do see nice range and stratification across these different domains. These lines are what are considered clinical cutoff for sleep impairment, if you will. So we're not seeing profound sleep impairment behaviorally, but we are, except for maybe a couple of these areas like daytime sleepiness. But again, we're seeing that this measure is able to be used in this population, and I think that's an important take home. So again, just want to show you the kind of things we're doing in this population, and I think that there'll be a lot more to say about this, but I'm hoping that this kind of work with the sample sizes we've been able to collect, based on the fact that we've been coming to family meetings and bringing these kids into the various clinics, I'm hoping this will be a model for us to think about outcome measures even in other syndromic forms of neurodevelopmental disorders. Okay, I don't have a ton of time, so I'm just going to tell you about TSC and skip the last one, because I really do want to tell you about this, because I'm hoping this is a model that we can use for DUE15Q as well. So I'm not going to spend a lot of time giving you the background on tuberous sclerosis. Many of you probably know about TSC, but what's important about TSC is it's an autosomal dominant condition, very highly penetrant for autism, but importantly is often diagnosed in utero. So we are able to follow these infants from birth. That's really the bottom line here. And so I had a study for several years where we actually did study infants with TSC from early infancy, and we asked whether there were early predictors of autism, because we knew there was such a high rate of diagnosis, yet no one had really studied these babies in early infancy. And again, all of this work is already published, but what we found was that as early as 9 months of age, we were seeing differences in social communication behaviors in our infants with TSC who developed autism. So these are the kids who developed ASD. They have higher scores on this measure called the AOC, particularly in things like eye contact, joint engagement, social referencing. And I only build this up because, and then we, because we're interested in brain function, we also looked at EEG biomarkers and found significant differences in long-range connectivity in our toddlers and infants who went on to have autism. So we had these, we had a biomarker, we had clear behavioral changes in the first year, and we thought we really need to do and study early intervention. These babies do not normally receive early intervention. The focus is largely on their epilepsy and maybe some global developmental issues they might have, definitely nothing focused on autism. And so we designed a clinical trial, and it was using a behavioral intervention called JASPER, which is a really well-studied intervention for social communication skills in infants and toddlers, and developed by a colleague of mine at UCLA. And so we designed this clinical trial. It was a big NIH R01. The trial was called JETS, Jasper Early Intervention in TS. And we were so excited about this trial. It received a lot of interest in the TS Alliance, in the TS community, nationally. It was 2 sites, Boston and UCLA. And the trial was set up so that we would basically see kids either immediately or after a wait list. So there was a wait list control design. And they would come in every week for intervention. And then we would see them at assessment time points to look at change, essentially. So we were very excited, and in the first year, this is what our map of enrollment looked like, which you can imagine, as it was at that time my first R01, and I did not want to give it back. And these yellow dots were the kids enrolled after 1 year. Our goal was 60. The purple was dots of families who were interested but did not enroll, and when we asked them why, they said, it's either too far for me to come every week or it's too much time. And again, these are infants who have epilepsy and other comorbidities, just like our kids would do 15Q. So what did we do? We had to go back to the drawing board, because when we interviewed these families, the issue was access. They couldn't get to us every week to do this intervention. And so we went back to the drawing board, and I'm not going to show these videos, but we basically turned the entire intervention into a remote intervention. And what we did instead was we brought the families in at one time point, at the first time point. They would get trained on Jasper. It's manualized, and there's like weekly program with it. I didn't mention it's a parent-mediated intervention. So the interventionist teaches the parent about interaction with the baby. Okay, so they learned the intervention live. We did all our assessments. And they would go home and they would practice. And every day we send them an automated text that says, did you get to practice? And we give them a choice, 0 to 5 minutes, 5 to 10. We're keeping it as easy for the families. They give us that information. And then and we send them home with iPads, so they get iPads. So once a week, the parent actually records their session with their child on the iPad. They upload it to a server that we developed on UCLA Box. The interventionists at each site download the video. They watch the session, and then they, through Zoom conferencing with the family, give them feedback, and then prep them for the next week's agenda, if you will, of what they're supposed to practice. So the entire thing turned remote. They do still ideally come back once a month for kind of in-person boosts. If they cannot do that, they at least have to come back for their assessment time points. So bare minimum, they're still coming back 3 times total, but that is much better than weekly, okay? So that was the change. 6 months, okay? So this was our map. And we actually just finished this paper, actually. We're writing this up because it was, I think, again, really important for the field of rare, when we think about research in rare diseases. But this was the map. 6 months later, this was our map. So the yellow dots, again, are kids enrolled. We now have like a wait list to even get screened for the study, because the families were so excited. They said, and we still have, families that don't enroll, and it's largely now because of the child's medical issues that make it hard for the parent to even think about learning sort of this new program, if you will. But outside of that, we've been able to mitigate the main issues to access, because the issues with these families, it's not about buy-in. It's not about being interested in research. It's about access. And so I think this kind of model is something that we need to be really employing across our rare neurodevelopmental disorders. Okay, so I'm not going to go into this because we're running late and I want to make sure we finish. But I'll just say that, we have a multidisciplinary clinic at UCLA where we see all of our kiddos who have either suspected or known genetic syndromes associated with autism. And in that, what we've been doing is collecting a lot of information about the diagnostic journey. So we interview families about their journey. And part of the goal of that, again, I think of it as clinical trial readiness, because the goal is to engage with our families and learn from them about what the challenges are. What are the things that we can do to make their experience better, if you will. The main thing I'll show you here is that when we ask them about barriers, and again, this also is going to be published. We just finished our final cohort, which is 40 kids. A bunch of them are kiddos with Do15Q. The barriers are, again, largely around access. Families are saying they can't access specialists. It takes a long time to get in to see a specialist, right? One I really want to emphasize is parents as advocates. So most of our parents were saying they were the ones who were out there trying to get the information on whatever their child's condition was because they felt like no one else had enough information. They were relying on patient advocacy groups for support, for education. 75% of our families were in PAGs, and the ones that weren't were because there was no PAG for their condition because it was so rare. So this is really important. And again, I think when we think about designing trials, making trials more effective, we have to partner with the PAGs because they're where parents are going, and it makes sense because they're really the source of all the information and support that our families need. Okay, so that's basically it. So take-home points, there's a lot of challenges in clinical trials and rare conditions, including, our sample sizes and patient selection, access to research. Our outcome measures are really lacking, if you will, and then we really need more quantitative measures of target engagement and change. I think we need to develop partnerships with PAGs, and we need to leverage technological advances. JETS is an exciting start, but it's clunky. I mean, we're like uploading to Box and using iPads and sending videos and Zoom, and I mean, some smart person and rich person could develop an app and make this really easy, right? And it will happen. That'll be the next iteration, right? But there's no reason why we can't be making this more accessible today. I mean, we shouldn't be having to go through so many systems just to make research easier. So anyway, so that's that. I'll stop with that. And just say, this is Carly. Abby does a lot of the EEG work in the lab. Joel is the one I was mentioning who did all the beautiful EEG work with Do15Q. And then Carly's actually at this meeting, and she's sitting there with a laptop waiting for families to upload those sleep EEGs. So she's been the coordinator on a lot of this work. And I'll stop there. Thanks.